Hello everyone. In this lecture, uh, we'll learn about antibody engineering. So, what antibody engineering is? Antibody engineering is the process of altering antibody structure and functional properties by recombinant DNA methods. Okay, so once if we can identify the sequence that encode the variable regions of the antibody, then by changing that DNA sequence, we can change the amino acid sequence of that part of the antibody. Thus, we can change the antibody. Okay, so how we can change the sequence of the DNA? By using various in vitro mutagenesis process. Okay, by using various in vitro genesis method, we can change it in different way. Like we can insert a extra gene, we can delete a certain portion of the gene, or we can do point mutation. We can change one or several amino acids. Okay, or even we can change the entire variable domains. Okay, so why we need to use recombinant antibody? Because the ability to manipulate the antibody genes makes it possible to generate new antibodies in vitro. Okay, and this can be done by making new combinations of heavy and light chains of the antibody, or by mutating individual complementarity determining regions. Okay, so if we can generate new recombinant antibody, it has various applications, such as we can use them for diagnostic purpose or therapeutic purpose to treat various diseases. Okay, now there are some key steps of recombinant antibody method. If we want to generate a recombinant antibody, there are few steps, such as first we have to identify the antibody gene. Okay, and then we need to do necessary changes in that gene. Okay, then we have to amplify the gene by PCR, then clone it into a plasmid. Then we have to use a expression systems. There are different expression system. I will talk about this later. Okay, and from that expression system, if we use there are different expression system, and that by using those expression system, we have to express our antibody. That means we have to produce our antibody, and from there we have to screen out or isolate the functional antibodies, and then have to select our antibody. Okay, so now let's talk about the expression systems. Okay, so there are basically three types of expression system. One, mammalian expression system, bacterial expression systems, plant expression systems. Okay, so next I'll explain these three different expression systems. First, mammalian expression systems. So in mammalian expression systems, mammals are used. Okay, say for example mice or rabbit. Okay, so this picture, it shows the use of mice to express our desired antibody. Okay, so we want to use mice to express our engineered antibody. Okay, say for example we want to express a human a human antibody by the mouse. So how we can do that? So first step is we have to isolate or identify the gene of the human antibody, such as for example these two genes. Okay, so we have identified the uh, variable, we identified the gene for heavy chain and gene for light chain. So once we have identified our gene, if we want to make any changes, we can do it by uh, genetic engineering, recombinant DNA technology. And once we get the gene, then we have to amplify it. Okay, so we have identified the gene, made necessary changes and amplified it. Okay, so we have amplified. So now we have enough amount of that gene. Okay, please keep one thing in your mind, that when we use the human gene, okay, we also have to include the C mu and CK gene along with the gene that encode the chain of the antibody. Okay, chain means heavy or light chain of the antibody. So why this C mu and CK gene is necessary? Okay, so by the rest of the antibody gene, different part of the antibody is produced. Different part means, for example, the heavy chain, the light chain, the variable regions, etc. Okay, so but after the different part of the antibody is synthesized, they need to be rearranged to form the functional antibody. So this gene product, I mean this gene product of C mu and CK are necessary for that rearrangement. Actually, gene product of C mu and CK do the rearrangement of the assembly. Okay. Okay. So then now we have to make a, I mean we after PCR or amplification we have got enough a gene. Okay. Now, okay, so keep that gene in your hand. Now we have to generate a transgenic mice. So what kind of transgenic mice? So first collect the embryonic stem cell from a mouse. Okay, then we have to knock out this, that embryonic stem cell. What kind of knockout? We have to delete the C mu and CK gene from that mouse, from that mouse cell. Why? Because mouse also produce different antibodies, right? So that means mouse also need the C mu and CK gene to assemble their antibody. Now our goal is we want to use the mouse to assemble our human antibody. So that's why we will knock out the mouse C mu CK gene. Okay, so that the mice will not be able to assemble their own antibody. Okay. Okay, so we have collected. So first collect the embryonic stem cell from the mouse and delete the C mu and CK gene from that mouse cell. Okay. So now then we have to transfect. So now we have the knockout ES cell, mouse ES cell. Then we have to transfect or transfer the human genes or human antibody genes that we have just amplified. We need to transfect them or insert them into that knockout mice cell. Okay, then we have to put this transfected ES cells, mice ES cells, into the mice blastocyst. So from that blastocyst mice will be born. Okay, so now as these blastocysts contain both mice cells and the knockout cells, okay, I mean the original wild type mice cells and the knockout cells, so the mice which will be born from this blastocyst will be a hybrid mice. That means chimeric mice. I mean some of the cells of that mice will express the human antibody and some of the cells will express the mice original antibody. Okay, so by using that chimeric mice and doing breeding, we have to get our transgenic mice. So that transgenic mice, I mean in that transgenic mice, all the cells they will express the human antibody. Okay, not the mice one. Okay. Okay, so once we got our transgenic mice, then if we immunize this mice with the antigen, which antigen? The antigen, I mean the which will induce production of this antibody. Okay, so if once we get our transgenic mice, if we immunize it with the antigen, then it will produce our engineered human antibody. Okay, so this was the mammalian expression system to express our engineered antibody. Okay, so now another expression system, the bacterial expression system. Okay, so this figure shows the example of E. coli. Okay, so E. coli expression system. Okay, so okay, how this expression system works? I mean in this expression system we use E. coli to produce our engineered antibody. Okay, so first what you have to do, say we have the transgenic mice, that means which produce the human engineered antibody. Okay, so we have to collect the spleen from that mice, because as spleen contain most of the B cells. Okay, so from that spleen we have to harvest the B cells or the hybridoma, then we have to extract the RNA. So now the extracted RNA will contains all the different types of RNA, messenger RNA, tRNA, ribosomal RNA, etc. But what we need? We need the messenger RNA, because the messenger RNA encode the protein, that means the antibody. Okay, so there is system by which the messenger RNA can be purified from the mixture of different RNAs. Okay, oligo-DT cellulose is such a material, okay, which can be used to purify messenger RNA. Okay, so after we have purified the messenger RNA, we can make the complementary DNA by using reverse polymerase reaction. Okay, by using reverse transcriptase. Okay, so now we got the cDNA which encode different antibodies. Okay, so now what we have to do, we can now choose or design different combination of primers which will amplify our desired or designed heavy and light chain. Okay, so we have to design, I mean, this is our choice. We can design different combination of primers. Okay, so by using those primers and using the cDNA as template by PCR method, we can amplify or make different heavy and light chain. Okay, then once we get the different heavy and light chain, then we'll insert them or clone them in different combinations into vector. Okay, so in this case the phasmid vector has been used. Okay, so that means by this process actually we can generate different phasmid vector which has the H and L in different combinations. So that means basically we can create a library of vector, and in each vector they they have gene of for different H and light chain. Okay, so by this method we can produce the library of phasmid vector. Okay, so once we have the phasmid vector, simply if I say simply we have the vector, then we transform this vector into the E. coli. That means we have to put this vector into the E. coli. Okay, then we have to screen out our transformed E. coli, that means the E. coli which contain this vector. Okay, so and those transformed E. coli, when they will grow, they will express this gene and they will produce the desired antibody. Okay, so once we get the transformed E. coli, we can basically grow them in large quantity, in large volume, okay, and produce our antibody in large amount. Okay, so this was the bacterial expression system. Now the another expression system, expression using in plant cell. Okay, so in this case we need to generate a transgenic plant. Okay, so if we can generate a transgenic plants which has the gene for the antibody, then whenever this plant will grow, they will produce that intact antibodies. Okay, so there are three major ways to make a transgenic plants. That means there are three major ways to put a foreign gene into a plant: by using agrobacterium mediated transformation, by using gene gun, or by using viral vectors such as tobacco mosaic virus can be used to introduce a foreign gene into a plant. Okay, this photo, it shows the agrobacterium mediated transformation. Okay, so what we need to do, first we have to take the agrobacterium and the TI plasmid. Okay, and we will clone our gene of interest, that means the gene which encodes the antibody, into the TI plasmid, and then insert this TI plasmid or transform into the agrobacterium. Okay, so then using this bacteria will transfect the plant cell. Okay, so once the uh, plant cell is transfected by the bacterium, it will have that plasmid into it, and the gene contained in that plasmid will get incorporated into the plant cell gene. Okay, so that means then we have to screen out the plant cell which contain that incorporated gene. Okay, so and from that plant cell, if we develop the plant, it will be the transgenic plant, because that plant cell contains the gene of our interest. Okay, so whenever that plant will grow, it will produce the antibody in it. Okay, so then we can do two things. One, we can extract the antibody from the plant cell, okay, and use them, or we can use that plant by I mean, if we eat that plant, okay, then we will get the antibody. Okay, so that means we can use it as a edible antibody source. Okay. Okay, so that's all for this part. Okay, so in the next part, I'll talk about the monoclonal antibody and their use in cancer treatment. So thank you for your patience.